Right, we are almost at the end of this meeting, and it is my pleasure, my honor, my everything to introduce Luis to you. He has been my PhD supervisor, so I know him since a very long time, and I can definitely say that Luis is one of the pioneers of the field of synthetic biology. So some, of course, of them were based in America or in the US, but actually then some were based in Europe, and Luis is one of those. And we heard during some of the talks in the past, for instance, the use of the negative feedback loop, right, to put in circuits and so on. And actually that was his first paper in the field of synthetic biology, showing that if you have a negative feedback loop, you have nice control over the amount of the target protein, and you have less noise and some homogeneous populations, so on and so forth. So I have to say that this was a study, is one of the first studies that showed how you can take engineering principles, put them in biological circuits. So Luis is actually a cell biologist, or a true biologist. He's not like an engineer or a physicist or something like this. So he really studied biology, and he had his PhD in Madrid, and then he moved to the lab of Alan Fair in Cambridge, where he's been postdoc for a while. For some years, and then he, very successfully, I have to say, and then he became a PI at the EMBL, so the European Molecular Biology Laboratory in Heidelberg, where he's been for many, many years, and then he became the director of the CRG, so the Center for Genomic Regulation in Barcelona. I don't know if he's going to show the picture of this institute to make us all very very envious of how beautiful it is just next to the seaside, where they're keeping on doing fantastic science. And he has been working on mycoplasma as a chassis for curing diseases, mostly in the lungs, as far as I know, for many, many years by now. And I think that this will be what he will talk about today, possibly, I don't know. And the floor is yours.
Thank you very much for your kind introduction, Barbara. It's a great pleasure to be here. So I will have structured my talk in two parts. First, I will explain a little bit the whole origin of the project, what we did at the very beginning with our back, and then I will present the most recent results we have got. And the next slide I will put is to show you sometimes how movies can influence the way you do science. Probably many of you are too young to remember this movie. It was an interesting story in which there was a scientist that was going to save the world. And the guy was dying because he had an incurable tumor. And the way to solve it was to take a submarine with some people and minimize it to the size of a cell, inject it, and then the guys were navigating in the submarine across the blood vessels, fighting with the red cells, the leukocytes, the macrophages, etc. And finally, they reached the tumor, destroyed, and also they were fighting with antibodies and all this kind of stuff. So then the idea came is why, and it was a long time ago, instead of making a mini submarine, we cannot use a living system for human therapy. And of course, in this case, there are people which are using viruses. We thought of engineering a bacteria as a living pill to do the role of the submarine in this case. That is, engineer it so it will go up to the place we like in the human body, recognize what is wrong, and react to it. So that's why it will be an intelligent pill and not just a drug that you take it and doesn't care if you are sick, not sick, you have fever, no fever, etc. It will just do whatever it does, independently of your body. And this is a field, of course, now there are many groups working on this. This is just a summary in a recent review we published in which some people are engineering the microbiota, and we heard yesterday very nice talks about microbiota engineering. This is one way of doing it. The other way is to take, for example, some pathogenic bacteria, modify them so they become non-pathogenic, and then, because they have preference to go to certain tissues, use them to treat different diseases. And I think the only one that is approved so far from therapy is an engineered strain of Mycobacterium tuberculosis for bladder cancer, because it stimulates the immune system and destroys the tumor. So the bug which chose to work with is Mycoplasma pneumonia. Oh, this is not working. Is that okay?
It does not work.
It was working before when the guy used it. So anyway, we chose this bacteria because at the beginning we thought that we wanted to have something simple to engineer. You get something very complicated, you don't understand 90% of it, so how you can engineer it safely. So we look for something small. Also, there was another pragmatic reason, that is, there was an old German professor in Heidelberg downtown that has been working with this bug for a long time, and he was retiring, so we could inherit all the reagents. So life is like this, no? So we chose this bug. It has 816 kilobase genome. When we started working with it, there were 680 proteins assigned. And talking about the small proteins, the reason why there were only 680 is because at that time, whenever you found an open reading frame smaller than 100 amino acids, unless it was found in other bacteria and other organisms, you just didn't annotate, because then you will annotate thousands of proteins. This bug has around 150 small proteins, many of them essential. Then over the years, we did a full systems biology analysis of this bug, so probably the organism for which a single group has collected the largest data set of everything you wanted to know. So we know, well, Mark there did the transcriptome, that is there. We know the copy number of every RNA, the copy number of every protein, the concentration of every metabolite, the structure of the chromosome, all the small open reading frames, the regulation of protein translation, essentiality, etc. So it's really a really huge amount of data. And at a certain point, continuing with what Barbara was saying about negative feedback modeling, etc., there was the time that Jonathan Carr and Marcus Kober published that paper in Cell, in which they published a whole cell model of Mycoplasma genitalium. And they used data from E. coli, which in a way is like trying to model human using E. coli data, because Mycoplasma genitalium and E. coli are more separate in evolution than humans and E. coli. But anyway, we collaborated with them because we had the real data from pneumonia. We made a whole cell model, and it was predicting the cell division time, etc. But we never published because at the end I was more successful predicting the effect of a mutation with my finger than with the whole cell model. So we left it frozen. Probably now that we have advanced, we could do more things. But at that time, because it's so simple, I could predict it without any model. I mean, what will happen if I will mutate something? So over the years, as I say, we collected all this information, and then we decided to move into the synthetic biology part, engineering it for treating human diseases. My original idea was to be there, understand it so much that I will be able to engineer it like you engineer the Airbus. You know now that when you engineer an Airbus, the Airbus gets out of the hangar and it flies, doesn't crashes. Well, yeah, we are not there. We understood a lot, but still not everything. But we thought, okay, if I will want to understand everything, I will die probably a couple of times before I fully understand it. So we decided to start engineering without getting all the full understanding of the bug. So, and this is something also interesting. When I chose this bug to work with, I never thought about containment. I never thought about secretion of protein for therapeutic purposes, etc. But at the end, we were lucky because this bacteria has a very interesting property. It doesn't have cell wall. Therefore, it doesn't have lipopolysaccharides. And therefore, it doesn't trigger massive immune response. Second, because it doesn't have cell wall, everything that it produces is secreted automatically to the medium. So for therapy, it's good because Lactococcus or E. coli or Salmonella, many proteins are stuck either before the cell wall or between the two cell membranes. Also, it has a natural biocontainment because it has a unique genetic code. There is a stop codon that encodes for tryptophan. So if you put a gene in this bug, it will not be taken by another bacteria because it will not be able to translate it if you use this stop codon for tryptophan. So, but I say, all these advantages, when we chose this bug, we didn't think about them. We were lucky afterwards to find that they has all of them. Of course, if now you When we started working with this bug, the only tool that we had at our disposition was a transposon, that it works very well. It does random insertion. But if I will go to EMA or the FDA and say, look, I have this nice bacteria that cures cancer, but I don't know where my gene is inserted, they will raise an eyebrows. So you would like to put your gene in a precise position every time in the genome. So you need to develop genome engineering tools, and over the years, I'm not going to enter in detail, but we have developed methods that allow us to replace or insert easily in one go up to 30 kilobases of DNA and to put genes wherever we want in the genome. Normally, we don't do this when we are testing new things because it's cumbersome, but if we want, we could do it. Then the other thing, of course, is you need to be able to design promoters because you would like to use promoters of certain strength. And there are several ways of doing it, but like everyone was insisting all these days, if you do things in the old way, one by one, so one promoter and another, and test and test and test, you die, so you need to find omics ways. And we developed this method based on the DAM methylase. Mycoplasma doesn't have DAM methylase. So essentially, you made a library of promoters that have several copies of the GATC motif before, and that they regulate those promoters, the expression of DAM. You just transform, divided the genomic DNA in two halves. One you digest with an enzyme that recognizes methylated GATC, and the other one that recognizes unmethylated. You do deep sequencing, do the ratio, and automatically you know how much DAM methylase is expressed in your bag. And when you take isolated cases, you see a beautiful linear correlation between the number of DAM protein copies per cell and the log 10 of the ratio. And it has an amazing dynamic range of 1,000. So with this, we could analyze hundreds of thousands of promoters and try to find which ones are the good ones to get a lot of expression, middle expression, low expression. What we found, okay, as I say, we found these promoters, but also it was a little bit sad, the regulation in this bacteria, because if you look at the position of the Prino box and you look at the preference for bases before and after, essentially the only important thing are the first 5 bases before the Prino and the next 3 bases, and that's it. Doesn't have a minus 35 element, doesn't have anything very sophisticated. And the other thing that was again by serendipity is something we found that later people have described it in E. coli and Bacillus subtilis. Is when we made the promoter library, the person that did it, I don't know if the night before was drinking with friends or not, but decided to make 2 libraries for whatever reason. One in which the first base expected after the RNA will start is an adenosine, and the other one that was starting with a thymidine. I tried to understand why. I discussed with the person. It was impossible to understand why. But anyway, we made 2 libraries. And then when we were sequencing the libraries, we found that on the left side, the majority of the transcripts were always starting with an A. But when we were using the other library, they were skipping the T, and they also tried to start with an A afterwards, and it's not an A, a G. So they didn't like a T as the first position of the mRNA. And then you realize that in bacteria, I don't know if in eukaryote the same, is that the RNA polymerase, the sigma factor, you open the DNA, the polymerase comes, and if the first nucleotide takes too long to come in, the RNA polymerase is released and you stop transcription. So the point is that the highest concentration in the cell is ATP and GTP. TTP and CTP are low concentration. So the most efficient way to start transcription is with an A or with a G, and this happened the same thing in Bacillus and in E. coli. So if you are designing a transcript, never put a T or a C as your first base, because you will have less RNA by definition. And then it was interesting because we were looking at all the ribosomal operons shown up top on the right, and I think we did like 200 perturbations, and whatever we did, all these genes were upregulated or downregulated at the same time. And I look at the elements before the Prino box, elements after the Prino box. Everything you wanted to look at in the wall, and we could never find anything that could explain why all these genes were co-regulated. But then if you look a little bit on the right, you see that all of them have GCT conserved. And I told you that the polymerase doesn't like to start with a C or with a T. So you are forcing all these transcripts to start with a G. Then in bacteria, you have this enzyme SPOT that when there is a starvation, converts GTP into PPGPP, that is an alarmon. So it decreases the level of guanosine. By decreasing the level of GTP, all these genes will be downregulated. And when there is food, GTP will go up, and all the ribosomal operons will be translated. It's a beautiful way to ensure regulation of many genes without using a transcription factor. And I think it's quite elegant what nature does sometimes in this respect. But normally, you will never look at the GC because it doesn't look like a transcription factor binding place or whatever. Then, of course, you need to secrete the proteins, and this is in this case, what we did is we cloned a series of signal peptides that we saw in the bacteria and fused many different proteins, and we found one signal peptide that whatever we put was very efficient in secreted proteins to the medium. And then the final thing is a defined medium, because if I want to use this bacteria to treat a human disease, it's not a good idea to grow it in rich medium. And this bacteria, to grow, you need 10% fetal bovine serum. And there is a big chance when you use fetal bovine serum that you could have viral contamination. So if you grow your bacteria in a medium with serum, animal serum, there is the chance that you will contaminate the patient. It happened some years ago with a pig vaccine that it was grown in also in a horse serum, and they infected the pigs with whatever virus that killed many of them. So you need to make a defined medium without any animal component. So for this, we look at the metabolic map of this bacterium that you see there. It's very simple, and this is an advantage to think about the medium. It's a disadvantage because you cannot hide anything. If you see the metabolic map people publish on Collie or human, no human brain will say if the guy is bullshitting or not, because you will not see if there are futile cycles, if things are going anywhere. Here you see where everything goes into everything. We found the things in orange are enzymes that are not there, but we know the activity is there because we see the metabolic products, and this happens in many organisms that there are moonlighting functions and probably other enzymes are doing this reaction. For example, something that is interesting is this bag doesn't have the enzyme that goes from nucleotide diphosphate to nucleotide triphosphate. So in theory, it should not be able to make any triphosphate nucleotide, but the pyruvate kinase does this reaction. It does it also in human. It's not very effective, but you divide every 8 to 20 hours like this bag, you don't care so much. So you don't need this enzyme, you just use the pyruvate kinase. Anyway, based on this metabolic map, we made a medium called BB13 that allows to grow the bag to the same density, takes a little bit longer, and doesn't need any animal serum. And then the final thing is this bacteria colonizes our lungs. We will have, many of us, we will have it in our lungs. As long as you are healthy, it doesn't do anything. But if you are immunocompromised or you get an injury in your lung, it will be able to grow and produce mild pneumonia. And this is, of course, not a good idea if you want to use it for therapy. So then you need to remove the pathogenic determinants, but then it's a balance. If I remove all the pathogenic determinants, it will be that it will not be able to produce anything in the lung because it will be dead, will not be able to eat. And if you remove too little, it will be pathogenic. So you need to start removing one by one and see what happens. At the end of the day, we develop this strain in which we then remove a toxin, a secreted nuclease, and the glycerol, the GLPD enzyme that produces peroxide and replaced by one that produces water. And we made this non-pathogenic chassis called CBH. I don't have time to enter, but later on we have modified the lipid composition of lipoproteins because lipoproteins in bacteria activate TRL2-1 and TRL2-6. And we have replaced a deacylated lipoprotein by 3-acylated lipoproteins that don't activate those receptors to even decrease more any possible pathogenicity. And what we saw is that if you look at the colonization in mouse of the wild type and the CBH, both of them stay the same time in the lung. Something that is very important, in a healthy mouse lung, after 2 days, there is no mycoplasma left, or in a healthy pig. So it's washed away. But if you have a lesion, then it can survive until 14 days. The reason is because it has a very poor metabolism. It cannot make nucleotides, it cannot make amino acids, so it needs to eat from the body. If the lung is healthy, it cannot eat. But if there is a lesion, there is cell debris, there is food, and it can survive. And this is good for therapy because it means it will stay as long as the tissue is not repaired. And TCBH, and as on the right, you have also a significant decrease of inflammatory markers compared with the wild type. The second thing is, as I was telling you, is, of course, I will have a non-pathogenic bacteria, I have a defined medium, I have the tools, etc. But now you need to show that you can secrete biologics in the lung. And over the years, we have tested, and every single cytokine, growth factor, transcription factors, nanobodies, single-chain FBs, FCs, etc., they are produced and they are folded and they are active. We only fail with IL-12. Interleukin-12, that is an heterodimer, for whatever reason we cannot. But all the others, they are produced and they are functional. But life is never perfect. This bug is good, as they say, it's small, we understand a lot, it secretes biologics without any problem, but it's small. It's 0.1 cubic microns. E. coli is 1 cubic micron. E. coli has 100,000 ribosomes. This bacteria has 500 ribosomes. So the amount of protein you can produce is not very large. So if you want to do therapy in the lung, you will need to secrete enough protein to have a physiological effect, because you don't want to overload the lung of the patient with our bacteria. You want to have the minimum number of bacteria applied and the maximum therapeutic efficacy. So for this, essentially, what you need to do is to combine this bug with protein design. So in parallel, when I was at the MBL, we started developing a software for protein design based on first principles that is called FOLX. It's used by many researchers over the world and companies. And this allows you to do mutations, design loops, antibodies, etc. And the idea was then, if I'm secreting proteins and I have protein design tools, now if I increase the activity 10 times, it means that I will need 10 times less bacteria to have a therapeutic effect. So at the end, the idea is to combine these two things. Our protein design software combined with the most recent design tools like AlphaFold or BOLS or MPNN. And we found that using Rosetta or FOLX, which are first principle force fields with artificial intelligence, is what gives you the best results. And this is the basis of a company we made for cytokine design. And then you combine with our mycochassis to secrete different proteins that will have a therapeutic effect. And at the end of the day, we want to create a therapeutic platform therapy integrating all these different things. Now, what I will go is just to finish the most recent data. I'm not going to show everything we are doing because we are secreting many different biologics with different activities, but I will give you some examples of what we are doing or what we could achieve with this bug. So when we started this project at the beginning, we were working with ventilator-associated pneumonia. So this happens when you go to the hospital and you are intubated. A significant amount of people, if you stay more than 2 days with intratracheal tube, you start having bacterial colonization, formation of biofilm, you get pneumonia, and there is, depending on the cases, around 20% cell death due to this. So we wanted to engineer our bug to first dissolve the biofilms, form on the intratracheal tube, because the bacteria, by making a biofilm, they become resistant to antibiotics, and to kill the colonizing bacteria. And the reason why we moved from here to the other two is because one thing is science and the other thing is market. We were developing this, and you saw the results are really amazing in mouse and pig. But when you go to investors to go to preclinical, they say, well, infectious diseases is not exciting. So you need to treat other diseases, acute diseases that you will die. And this is why we chose these two that I will explain later. So here the first thing we did is, one of the first things we did was to express one of the problems, again, with the infection with Pseudomonas aeruginosa is that it causes massive inflammation. So we thought that by expressing IL-10 in the lung, we could alleviate the inflammatory response to Pseudomonas. And we wanted to express IL-10. IL-10 is the most typical cytokine with anti-inflammatory properties. Also, like every cytokine, under certain conditions, it can be pro-inflammatory, and now some people are using it to treat cancer, but this is life. The problem with this molecule is a swap domain. A swap domain in a protein is when you have a protein like this, and then it opens, and then the same monomer packs against the first one in an anti-parallel fashion. So it's a dimer of two similar monomers in which the domains are open. So what we did is to design a linker in red to make the swap domain a single chain, and then we introduce mutations using our protein design algorithm to improve the activity of the mutant single chain between 50 to 20 times compared with the wild type. And then we developed this mouse model infected with Pseudomonas PAO1 strain, and you look at some inflammatory markers, and you will see, for example, when you infect with Pseudomonas, tumor necrosis alpha or ILH, they go up. If you apply now the wild type IL-10 secreted by our bacteria, there is not much effect because IL-10 is not so potent and we are not producing a lot. But when we are producing the improved mutants around 50 times, then you can see that inflammation disappears. So the idea of making your protein more potent and combining with bacteria, this works. And then, as I say, we decided to treat the the pneumonia caused by Pseudomonas aeruginosa in mouse, in pigs, and also we work with intratracheal tubes from patients. And just with only one dose of our bacterial mycochassis expressing three proteins, a combination of glycosidases that dissolve the biofilm of Pseudomonas, and then a pyosine that kills Pseudomonas aeruginosa PAO1. This for me was an interesting discovery because we think that humans we are the only species that we kill each other based on our color, religion, or beliefs. But Pseudomonas species, they kill each other. They develop specific pyosines and vaccines against the pyosines, and one Pseudomonas aeruginosa strain will kill the other, and the other will kill another one. So there is a very nasty competition between Pseudomonas aeruginosa strains, each one having their specific pyosine and their specific vaccine against it. And yes, with one application of mycochassis, only one application, we got 50% reduction in cell death in the mouse. If you have two applications, essentially the mouse don't die. They dissolve the biofilm and they kill Pseudomonas aeruginosa. And also when we took intratracheal tubes of patients with ventilatory-associated pneumonia, and we treated them with the latest antibiotics or with our bacterial chassis and the latest antibiotic, you completely eradicate the bacteria from the tube and the biofilm, so you get it clean. And then we did the same thing in pigs, and also we found the same effect that our mycochassis expressing the drugs against Pseudomonas decreases the content of Pseudomonas aeruginosa in the lung, inflammatory markers, etc. And I say, at this point, we were ready to go to preclinical, but then to go to preclinical, you need several $1 million, and then the venture capital decided that it was not interesting because there were other possibilities to treat BAP. So then we moved to these two other diseases. Idiopathic pulmonary fibrosis is a very nasty disease. No one knows really why you get it, but from the moment you are diagnosed, you have maximum 2 years of life unless you get a lung transplant. There is no solution. The lung will stop working. And the other one is lung cancer and metastasis. So both are acute diseases, and we thought that they will be interesting targets because if you are dying, you will not say no to have some bacterial if it will have a chance of saving your life. If you are not going to die, then you will think twice if you would like to get a bacteria in your body or not. So the thing that we know about lung fibrosis, looking at chemokines, can be shown in this slide. It's a very complex diseases in which cytokines and chemokines and growth factors like TGF-beta play a very important role. But essentially, there are some cytokines which are bad and some cytokines which are good. And all the ones that have an asterisk, we have expressed them in our mycochassis, and we can express them functional, and we are testing all of them individually or in combination. But I will show you just 2 examples of this. The first one is interleukin-1 beta. Interleukin-1 beta is a bad one, so you need to develop an antagonist. There is one known called isunakinra, developed by Bushard Therapeutics. And what we wanted is to improve this 1 beta, as we did with IL-10, so to have efficacy with our mycochassis. So again, using our software, we made a mutant that is 5 times more potent than the wild type. This is the fold change of our quadruple mutant versus the wild type. We determined the crystal structure, which is almost identical. The small changes in green are due to crystal contacts, not to conformational changes in the protein. And then we tested first of all, in the mouse infection model with Pseudomonas aeruginosa. IL-1 beta antagonist should be anti-inflammatory. So we look at some chemokines, IL-6, that is a pro-inflammatory cytokine, or KC, or another chemokine. When you apply Pseudomonas infection power 1 with our mycochassis CBH expressing the quadruple mutant, you see that in all cases you get a very significant reduction of inflammatory markers, which become closer to the PBS administration. Then by this we checked that our quadruple mutant was better to fight inflammation, and then we move into a fibrosis model. And the point with mice is that you need to develop a model that mimics the human disease, and sometimes you get approximations because there is no real model of lung fibrosis in mice. So the way to cause lung fibrosis is by applying bleomycin. Bleomycin is an antibiotic that does DNA damage, and for whatever mysterious reason, when you get bleomycin in the mouse lung, it develops fibrosis. But it's a fibrosis that after 2 months, it disappears, so the mouse don't die. So it's a temporary fibrosis. To get it permanent, you need to give several applications over a long time. In any case, this fibrotic model in mouse has an inflammatory phase. That is the one that we could fight with our engineered interleukin-1 beta, and a fibrotic phase. And what we look here is at 2 types of parameters, the air content and the fibrotic lesion. The one that perhaps is more clear is the fibrotic lesion. So you use CT to follow the lung with the mouse alive. And when it's black, it means the lung is happy. When it's white, it means the lung is not very happy because you have fibrotic lesions and you don't have air content. And if you compare the lung with bleomycin, especially it's typical in bleomycin, the left lobe is more affected. You have all this region disappearing and this region. With our mycochassis, without expressing anything, is the same, and expressing our interleukin-1 beta, you can see how you can recover the upper and lower part of the lobe. And this is shown then for several mouse. And at the day 21, out of 8 mouse treated with our mycochassis expressing IL-1 beta, 5 were exactly have fully recovered like the wild type. So we have shown that we can have a therapeutic efficacy in the fibrotic lung. And also we did transcriptome, and we saw when we treated the mouse with our bacteria expressing this interleukin-1 beta quadruple mutant, you see all these fibrotic markers go down and become quite normal. By looking at the transcriptome, what we see is that we are blocking the appearance of myofibroblasts that are the main culprits in lung fibrosis. Okay, so as I say, here we are expressing a cytokine IL-1 beta for the inflammatory phase. What about the fibrotic phase? And for this, you can use IL-22 because it blocks TGF-1 beta signaling that is involved in the fibrotic phase. And here is the same thing. We took IL-22 wild type, and then using Foldis, we designed mutants, and we made this mutant called IL-22AM that is 25 times more active. And IL-22 is inhibited by a soluble receptor, IL-22BP, and we found also that this mutant is not inhibited by the soluble receptor of IL-22. And then to cut a long story short, we tried in mass. We did 3 applications, and then we look at the fibrotic phase by micro CT, also look at markers, etc. But the most important thing is, if you look at the recovery of the mouse non-treated only with bleomycin and nothing else, we see 0% recovery at day 21, but the one treated with myco-expressing the engineered IL-22, 43% of the lung recovery with just these 3 applications. So it means now we have 2 different cytokines, one for the inflammatory and one for the fibrotic phase, and we are trying many others, and also chemokines, which are more complicated. Jamil in the audience knows about it. But the idea is to, of course, combine some of them for a combined therapy. And just to finish, also, I will give you a little touch about lung cancer and metastasis. Lung cancer is one of the leading death causes worldwide. And the first thing is, you want to treat tumor, you need to show that your bacteria localizes in the tumor, because you don't want to secrete biologicals in the healthy tissue. Here you have a lobe. This is the lung of the mouse. I think it's the right lobe, and you see the tumor in the center. And the brilliant dots are mycoplasma. So if you apply mycochassis to the tumor in the lung, it's washed away. You see that the lung is black, except in the tumor where the bacteria tend to concentrate. Only problem, it tends to concentrate in the periphery so far. We have not seen it getting inside. And then, what is the advantage of live biotherapeutics? It could be used to remove systemic toxicity. Someone before was saying that biologics, I think you were saying that biologic is a blooming market, that many pharma companies are using them, but many biologics fail to go to the clinic because you have systemic toxicity, which means, for example, interferon gamma is really nice because it kills the tumors, but if you have an injection of interferon gamma, you have a big chance of dying, not because of the tumor, but because of the interferon gamma. Has many, many targets in the human body. And this happens the same thing with many biologics. So ideally, you would like to have, for example, a bacteria that will deliver the interferon gamma only in the tumor and not in the rest of the body, and see if you can remove the tumor that way. Then you will avoid systemic toxicity. Also, we are working in masking the biologics so they are only active in the right tissue, but I don't have time to talk about this. So then the idea, first of all, is again we want to improve interferon gamma to express with our bacteria. Then we need to characterize that it's more active using, first of all, commercial egg blue cells to see that your interferon gamma is more active, and then macrophages or tumor cells to see that is functional. And finally, you will need to test it in a mouse model. So the first thing we check is that mycoplasma expressing interferon gamma. I forgot to say interferon gamma is like interleukin 10. It's a swap dimer cytokine composed of 2 monomers. So this is the monomer, and it's expressed very nicely. But then when we tested the monomeric protein in egg blue cells, so they become blue when they are activated by interferon gamma. The interferon gamma produced by the bacteria is pitiful. It's pitiful means 100 times less active. It's really not very exciting. You will not be able to do anything with it. So then, we go to protein engineering. We made a single chain, like with interleukin-10, engineering a loop between the two monomers. Then we got the expression of the single chain. And then when we look at the activity, now we have an interferon gamma that is 5 times more active than the wild type, just by making the single chain and putting together the two monomers. And then we made also some mutants, and I don't have time, but also we introduced some mutants, so we have now an interferon gamma that is around here, so it's like 50 times more potent than the wild type interferon gamma. And then we tested it. First, we tested it with macrophages, so when you apply interferon gamma, you go from a resting macrophage to an active macrophage. And then you are looking at the markers for the active macrophage, and you can see that the single chain with the mutations in green compare with without interferon gamma, or with a single chain interferon gamma without the mutations in yellow. You have a very nice activation of macrophages. The same thing for arresting cell tumor growth. You can see that you have a significant reduction. But the most interesting result, and now this we have fully confirmed, is we have not yet entered in lung cancer, but you made a mouse breast cancer model, and you see the growth of the tumor with time. If you apply only our bacteria and PBS, the tumor continues growing. And if we combine mycochassis with an antibody against an immune marker, something that activates the immune response, or our bacteria expressing our interferon gamma with this nanobody that stimulates the immune system, in 100% of the mice, you kill the tumor disappears, and there is no systemic effect, no lost weight of the mouse, etc. Of course, as everyone says, cancer in mouse has been cured many times, but still is a very interesting data that by locally secreting interferon gamma combined with an antibody that stimulates the immune system, you fully remove the tumor just by applying mycochassis in the tumor. So now the next step is to move into the lung and see if we will have a significant effect or not. And with this, I will finish. I mean, I just show you 3 examples of what we are doing. We have many others, but that's just to give an idea of what we are trying to do. And I think for me, at the end of the day, and I was discussing before, I could have said that we thought about everything from day one, and everything was according to plan and following engineering rules of synthetic biology. But in reality, it's a question of luck, serendipity, and some thinking that at the end of the day, you get something that seems to work. And these are the people involved in my group and the financing, and thank you all of you for your attention.
Questions? Yes, Mark. Comes the microphone.
Okay, yeah, very nice talk. I was curious about this cytokine design. So because of the topology of this kind of inverted dimer, so what do you do with the second part? Do you need to reverse it or something, or how is it designed?
No, when you make a single chain, then you can put mutations on both sides of the dimer. They are symmetric.
So, but you don't need to revert N-terminal to C-terminal or things like that?
No, you just link one end and C, you leave the other end and C open, and then you will have a single chain with the 2 molecules that fall very nicely, and you can introduce mutations. I mean, this we have done with interferon gamma, with IL-5, and with IL-10, and in all the cases, you get a more potent molecule than the monomers.
David.
That was a great talk. It's great that you're getting so much efficacy by putting in a few mutations into these cytokines and getting 20 or 50 times more efficacious proteins. I'm just interested if you have any thoughts on why those mutations aren't sort of naturally evolving, given that they're so much better proteins.
Well, I mean, you know that nature selects to the point that is functional and then stops selecting afterwards. A long time ago, when I was doing my postdoc here in Cambridge, we were working with Barnase, the ribonuclease, and then there is another enzyme called binase that had like 15 mutations in 110, so they are almost identical. It was 15 out of 110. Then I mutated one by one from Barnase to binase in isolation, and I found 4 mutations that made Barnase more active, 4 mutations that make it more inactive, and the rest neutral. And if you look at the combination of all of them at the end, both enzymes are similar. But if I take only the 4 good ones, I could make Barnase active like this. So essentially, in nature, probably you get a random mutation that is slightly bad, then the next random mutation should be slightly good. Otherwise, you will go below a threshold in which it will not be functional. But there is nothing that favors you to make it more active than you already did. Probably will not be good, healthy for us if we have a too active interleukin-10. But if you are doing it for therapy, you don't care so much. So I forgot to say that if you want to be successful in protein design is use a force field, AI, whatever, and always select those positions which are not conserved in evolution. That is the sacred rule. If you don't have any other choice, mutate something conserved. But if you can, just do a multiple sequence alignment from koala to human, see which positions are variable, and then those are the ones you can introduce mutations.
I was curious why, so you didn't really mention which antibiotic you used when you did the first application that you had with the infection, the one that unfortunately did not really attract the investors. Because if you can use, you were using, was it vancomycin? Is it the last resort?
I don't remember.
Kind of
We used the last resort antibiotic that is used when people are not responding.
Exactly. So, but could you not show, because then you used the same, so that was already quite effective, and then you had together with your mycoplasma chassis, and it was completely eliminating it, but at the same time the other one, so even just antibiotic was quite good. But could you not show that if you have your chassis and you take an antibiotic that typically does not work, but now you have your biofilm degrading activity and so on, then that one can be used, so you don't need to resort to this last resort
For
antibiotic.
sure,
that would be attractive, right?
But the point, well, I forgot to say that one big advantage of this guy, which also I never thought about it when I was started working with it, is because it doesn't have cell wall, the majority of the antibiotics don't kill it. So only macrolides kill mycoplasma. So you can combine with antibiotics and then have this combined effect like with vancomycin. But the main problem is it should be when there is no other solution. There is no, otherwise no one will take a live bacteria.
Yeah, but if you
because think of the cost. If you want to give a live therapeutic in a hospital, you need to have an isolated room, you need to be alone in the room, you need to prevent it going to other patients. This is very expensive treatment. So if there is any other solution, no one will use. This is the message I got from the investors.
Yeah. Well, it's just, I was thinking that there is, all the time we always read these papers about, okay, there is a need for new antibiotics, because there are no antibiotics working, so if you have a solution for that.
To pharma. I remember having a chat with a big pharma company, the CEO, and he told me, we have many drugs that could cure diseases in the third world, but there is no market.
I see.
It's like this, unfortunately.
Yes, a question here.
So I was wondering if you ever explored like dynamic delivery, like some kind of sensing system that might reduce like collateral effects or like secondary effects or anything. So you have tested like just constitutive delivery for your proteins. Have you ever tested like sensors that detect, I don't know, some cancer metabolite and then trigger the expression?
We are thinking of secreting kallikrein to digest kallikrein. That is one of the things also that blocks the immune response, for example. Also, there is one in the group. Myco has another advantage I didn't mention, that it produces vesicles because it doesn't have cell wall. Also, E. coli can do it, but Myco is very easy. It can make exosomes like a eukaryotic cell. And now we are delivering RNA and other molecules to the target cells. We are improving the vesicles to improve the fusion, the delivery, etc. But yeah, playing with metabolites, we were thinking, for example, of developing a lactate sensor for tumors, because normally they acidify the medium, expressing lactate. So then to activate expression in the presence of lactate, but something don't work. We couldn't get the E. coli lactate sensor to work in myco.
And for example, do you expect like the transcriptional regulation to trigger in enough time in the patient to be able to produce enough therapeutic, or?
So what if we get?
If you're like, in the end, if you activate with a lactate sensor, it's going to take some transcriptional time to then produce the protein and so on. Is the bacteria enough?
We did in vitro, not in vivo, the lactate sensor. We never could get it working for whatever reason.
Thanks.
Yes, there is another question there. I guess then after this question, probably we need to move on. Yeah.
Hello, thank you for the talk. So it's out of curiosity, we know a lot of people with mammalian cells are really cautious about the use of mycoplasma, and I suppose your lab will use a lot of mycoplasma with actual mammalian cells. Just wondering how the procedures, how the workflow will look like to ensure this kind of like things.
Well, normally when you mention to your colleagues at CRG that you're working with mycoplasma, they don't look at you with kind eyes. let's put it that way. We have a special room, an S2, separated from the rest of the wall, so not to contaminate, because you know, as you say, one of the biggest problems in cell culture is to have mycoplasma infection, and then you get interesting results that have nothing to do with reality, but with the infection of mycoplasma. Happens in the best families. But say we have a separate room, completely isolated from the rest of the wall. But this is what I'm saying, that you want to do therapy, even if we consulted FDA and EMA, what do we need, and even if we demonstrate in pigs and mouse that is not pathogenic, that doesn't cause lesions, etc., the treatment is going to be, again, very expensive, because you will need to treat the patient isolated, all these kind of things. So it only makes sense if you are treating diseases that there is no alternative. Either you get it or not.
All right, then thank you once again for this wonderful talk.